those two organelles that have to do with cellular energetics. So there's two, obviously, I hope it's obvious, two cells. You have the mitochondria, or organelles. Mitochondria has two membranes, as we discussed in the previous video that we just showed, as the ninth graders went over. And that's mitochondria. Bless you. And then the other one was the chloroplast. I'm going to go over the basics today, and then we'll go into details tomorrow. Control points. Where is there allosteric inhibition? Where are the control points? As I hope you know by now, after endosymbiosis, you know that the mitochondria is found in every eukaryote. The chloroplast is only found in plants. Plants have both mitochondria and chloroplast. So when you're looking at cellular respiration and photosynthesis, they obviously feed into each other. The overall reaction is simple, but the details are difficult. So when you C6H12O6 gets added, put together with oxygen gas in a system, in a cell, this is aqueous. What you yield is water. You make water, and you also make CO2, and obviously you make ATP. At least I hope it's obvious. That's the whole point, is to make this ATP. This is the goal. To run it in the opposite directions, here you're taking the energy that's here in a sugar molecule, and you're taking the energy and you're releasing it, and you're putting it into ATP. So if we were to take this yellow and transfer it here, you'd say the energy that was in sugar went into ATP. So the key is to try to get that energy out and put it into ATP. Now, obviously this process in this direction, using oxygen, is called respiration. Now, respiration would not be adequate because respiration also means breathing, but this is cellular respiration, which is the reason you breathe. The only reason you breathe is to get oxygen. But the only reason to get oxygen is so you can do this, to make ATP. ATP is the whole reason you're doing a lot of what you do. Now, to run the reaction in the opposite direction, for photosynthesis to work, you've already put all the energy to make that you had in sugar into ATP. The missing part in this equation that you don't see, that's incredibly important, obviously, at least I hope it's obvious, if you're going to run in the opposite direction, you're not going to use ATP to run in the opposite direction. That would defeat the purpose. And because of the first law of thermodynamics, since you're only able to get 10% every time you change energy, anytime energy or matter changes, the best percentages is 10% of what came after. You can only capture 10% on average. Natural systems only capture about 10% of what is available, of energy that's available. So it would be self-defeating to take sugar, break it up, make ATP, and then use ATP to make sugar. What's the point? It wouldn't be any. So the process of photosynthesis uses this energy source called light, hence the word photo. And synthesis means make. And you take CO2 gas and you add it to water, which is why plants need water. It's the only reason, one of the many reasons plants need water. And they produce oxygen and ultimately C6H12O6, glucose in aqueous solution, and oxygen gas. So that's the overall reaction, but obviously things are a lot more complicated. Otherwise, we wouldn't be here, and you would have gathered this already from elementary school, because they probably taught you something like this in elementary school. The two reactions are opposite. Please note that unlike what you've been taught in the past, plants do use oxygen. They not only make it, they use it. They have mitochondria, so they use oxygen. The mitochondria uses oxygen. So if you're running cellular respiration, that is oxygen. So if it's going to be cellular respiration, I hope it's obvious that we have to start with a cell. So let's start with a cell. As soon as glucose comes in, you have to trap it inside, because the glucose channels will allow glucose to come in and out. So you want to trap it inside. The glucose channels only go in certain cells when insulin is present. We went over that. When this glucose sugar monosaccharide comes into the cell, the first thing you do is you have to do glycolysis. Let me see if I have a... We look at glycolysis. What you're doing is you're taking glucose, and let me break this down. Glyco means sugar, or it's short for glucose. Os, the ending for enzyme or for sugar. Lysis means to break. So you're breaking sugar. Count the carbons, and at this point, everyone should know that glucose has 6 carbons. Count the carbons. Probably the word you're going to hear the most, besides hydrogen bonding in biology, is carbon, 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 carbon. So glycolysis is taking glucose, which is 6 carbons, C6H12O6. And you're going to break it down into 2 pyruvates. Those of you that understand some simpler arithmetic, some division, and some addition, how many carbons do you think pyruvate has? 3 is correct. So these are 3 carbon molecules. Started with 6, you end up with 3. This is all in the cytoplasm, in the liquid part of the cell. The fluid part of the cell, plasm meaning fluid, cyto meaning cell. So now you have 2 pyruvates. If oxygen is present, and that's a big if oxygen is present, then the 2 pyruvates will find their way into the mitochondria. That's a big if. If oxygen gas is present. If not, no oxygen, then what happens is it goes into a process called, again, this out here is step 1, which is glycolysis. Glycolysis, breaking sugar up. That's going to happen. Do you need oxygen to break sugar up? No. Glycolysis is anaerobic. Glycolysis is anaerobic. What does an mean? Does not need oxygen is correct. An means not. Aerobic means it needs oxygen. So this is anaerobic. Does not need oxygen. You do not need oxygen to take sugar and make 2 ATPs. By the way, you have a net. Remember that word net. You make a net of 2 ATPs when you do this. And get 2 pyruvates. That's not a problem to break sugar down. In fact, that early cell that was running around in the primordial soup, eating things, it did glycolysis really well. What the cell that it ate and did not, that mitochondria, what it can do is take pyruvate and break it down further. What it can do, and it was able to do well, was take and break down pyruvate using the Krebs cycle and the electron transport chain to get 36 ATPs out of each sugar. And I hope that you can see that that's why our earliest ancestor cell was able to outcompete all those other cells, because instead of just 2 every time it ate some kind of sugar molecule, it was able to make 32, instead of 2, it made 36 for every single sugar molecule it ate. But when there was no oxygen present, and that explains why the early cells only did glycolysis. Was there a lot of oxygen present in the early days? No, there were not. No, there was not. So since there wasn't a lot of oxygen present, doing glycolysis made sense because there wasn't any oxygen to be able to do this. Only later, when oxygen started showing up, did cells develop the ability, again through random mutations. I don't want you to think that I'm saying that somehow they made this decision. No, they didn't have a group of cells get together and have a conversation. The idea is that randomly these mutations occurred, and there's this prokaryote that was able to take pyruvate and get that energy out of pyruvate that the early cells were leaving on the table. This anaerobic condition of the earth early on made glycolysis the way to get energy. They moved around real slow, and that's fine. But if there's no oxygen present, you got to get rid of the pyruvate somehow. Otherwise, it's going to build up. It's going to be untenable. Also, you'll see when we start going over the details that you have to recycle some of the NADH, FADH2 electron transporters. So you had to do something called fermentation. Now, an easy way for them to test you, by the way, this is 2, process 2. Process 1 is glycolysis, which happens with or without oxygen. Glycolysis. Without oxygen. Fermentation also requires no oxygen. In fact, that's the only time fermentation occurs is when there's no oxygen. Bacteria and yeast being one of our earliest ancestors, is split off from an even earlier ancestor long ago. Yeast can do fermentation really well. so can humans. We do fermentation. Just about every cell does fermentation. There's a difference though. Fungi, let's just say the word yeast. Yeast are a type of fungi, single-celled fungi. Yeast do fermentation and they produce alcohol. All right, and CO2. They take the rest of that pyruvate, they make CO2. Then if you want carbonated champagne with some alcohol in it, take some grapes, usually white grapes, throw them in a bin, chop them up, smash them, get that sugar in the water, add some yeast, they'll turn into alcohol and produce bubbles. Why do we have these computers open, everyone's off task? What's going on with the computers then? Why are they open? All right. So alcohol and yeast are producing, and we're producing alcohol and yeast. Alcohol and CO2, that's what the yeast produce when they do fermentation. So they can recycle the electrons. Doesn't give them any energy. In fact, the alcohol ends up killing all the yeast in the wine. So when you drink wine, you're not eating any live yeast. They've all long been dead. That's with any alcohol production. That's what happens. Same with bread. When you cook bread, except you burn off the alcohol, the CO2 makes the bread do what? Rise. It makes little holes in the bread. When you slice bread, you can see the holes where the yeast used to be, and it produces those little cavities. So in any case, that's what yeast do. Some bacteria do the same thing. You could use bacteria to do the same thing. So bacteria and yeast will do this. We don't do this. Otherwise, we'd be drunk every time we went for the long distance runners would love it, I guess, because they could do long distance running and get drunk while they're running. That would be, I guess, interesting. But that's not what we do. Humans, that's right. Humans produce lactic acid as a way to recycle that NADH and FADH2 and also CO2. So we do expel some CO2 there. But lactic acid is the key so that we can keep this going on. This is a temp for humans. This is not enough ATP. For yeast, that's enough. 2 ATP for every sugar. Plenty of ATP for a single cell organism, especially if it's doing a lot of it. But we cannot survive on this. We will die. That's why if somebody starts choking you out, initially you won't die right away. If you start drowning, initially you're not going to die as soon as you hit the water, right? It takes minutes, in some cases 5-10 minutes, for death to finally occur. Brain damage might start after 3 to 5 minutes, depending on your size, how cold the water is, all that. Why does temperature have anything to do with it? Because if the water's ice cold, there have been people that we found underneath ice frozen rivers. They were technically drowned for 10-15 minutes. They survived with very little brain damage. That's because it was so cold, their body process required very little oxygen. So they were in a sense, they were in a type of hibernation, type of suspended animation. But what happens is you start to produce lactic acid, you start to make CO2, you make some ATP, but not enough for your cells to survive. If you go for more than a few minutes under normal room temperature, you're going to start to get cells dying, especially the most sensitive cells in your body, your neurons, and so you start to get brain damage very quickly. But after 5 or 10 minutes, every cell in your body is going to start to die. So your heart will certainly stop. Since it takes a lot of ATP to pump blood, your heart will stop very soon after running out of oxygen. Yes?
So if the pyruvate doesn't go to the mitochondria, then we do fermentation for lactic acid, and then bacteria do fermentation for alcohol?
That's correct. So remember, bacteria don't have a mitochondria. So just remember that. Bacteria, I know I put bacteria on here, but as a note, the only real thing that's going to have, the thing that has the yeast, the thing that has the mitochondria is the yeast. Yeast is a eukaryote. The fungi is a eukaryote. They have a mitochondria. They're not going to use the mitochondria if there's no oxygen. You don't use it. Mitochondria can't do anything without oxygen. It needs oxygen. Oh, is everything clear so far? Because these are some of the tricky questions that I've seen repeatedly throughout the history of the AP biology exam. I've seen come up over and over and over again. What's the last electron? Why is oxygen necessary for the process? When you add cyanide, when someone takes cyanide, how does it kill them? What happens when lactic acid builds up? Where does fermentation happen? Where does glycolysis happen? Do you need oxygen for fermentation? Do you need oxygen for glycolysis? Why do I still have computers open and people using computers while I'm explaining something I do not understand? Jonathan, would you like to explain why the computer's open? Your hand is continuously tapping on the keyboard. Huh? All right, close it and pay attention to what I'm talking about. So humans, bacteria, yeast, this is all happening all in order. So glycolysis happens no matter what. It does not need oxygen. You get the end product is, in general, is 2 pyruvates, 2 ATP, some NADH or some high energy electrons. And fermentation only happens when there's no oxygen. If there's oxygen present, it goes in the mitochondria. That's one control point. That's one simple control point that's based on whether there's oxygen or not. Easy way for them to construct. I could construct 5 or 6 complicated questions with just that, without any more details about enzymes, which enzyme does what, and product. When we look at the 3 steps, when there's oxygen present, right? Cytosol is another term for cytoplasm, all right? Cytosol. I'm not sure if there's a slight difference or not. Glycolysis produces pyruvate. How many pyruvates? 2 pyruvates for every glucose. The net production of 2 ATPs. Then you get NADH produced here. That gets transferred over to the electron transport chain. These electrons at each step get transferred over. That's the whole point, is to get the high energy electrons out of sugar and into ATP. Yeah.
The cytoplasm is the cytosol plus the organelles, and then the cytosol is just the liquid.
Thank you. So that's a good. I keep forgetting that, so I appreciate that. So cytosol is just the liquid. Cytoplasm is the organelles, and the liquid is what I'm hearing. So the pyruvate comes into the mitochondria. The first thing it does, by the way, is this is where you make CO2, right here. When you breathe out, everything you eat, unless you make it into fat, all the sugar you eat is turned into CO2. You breathe out your food. Everything you eat that you don't poop out or don't turn into fat, all the sugar you eat gets turned into carbon dioxide and you breathe it out. So you should not gain any weight if you're eating just what you're using. Theoretically, physicists say this all the time. It's not true because it gets a little more complicated. But it is true, but it's also a little more complicated than that. But bottom line, the matter that you eat, you will breathe out. Eat a tablespoon of sugar, you will breathe out a tablespoon worth of CO2. Same amount of carbons go out that came in, assuming you're using all the energy that you're taking in. That pyruvate, the 2 pyruvates come in, all the 3 carbons for 2 pyruvates, all 6 carbons get turned into 6 CO2 molecules. They leave, the electrons get sucked up. You produce 2 ATPs at this stage as well, and those electrons come in the electron transport chain that we'll go over all this starting tomorrow, and you'll produce a lot of ATP here, 25. Add them together, you produce about 25. Altogether, you get 32 or 36 ATPs, depending on how you count it, and how many you end up losing or gaining. That's the general steps. to the process. So it's, I think, a better, a neater version of what we just talked about. I said it can be as little as 32, it can be as high as 36, but they say 34 to 36 in general. If you completely oxidize all the sugar, you'll make 36. But sometimes things don't go the way you think. So why is the oxygen used? As you'll see, and I think some of you might remember, it's the last electron acceptor. If you don't accept the electrons at the end, then everything backs up. It's a giant conveyor belt. If you back up any blockage in the conveyor belt sends everything into chaos. This whole thing stops and everything gets backed up. All the pyruvate goes in this direction. If you're a mammal or a lot of eukaryotes, you're going to produce lactic acid. If you're in a yeast or you're a bacteria, you're going to produce ethanol. All right, and CO2, by the way. You do produce bubbles. The way they get you on these problems is they'll set up a scientific experiment where you measure the rate of photosynthesis, the rate of cellular respiration, the rate of fermentation by how much gas you produce. How many bubbles do you produce? And they'll ask you, what is the gas that's being produced? Well, if it's any of these processes, the gas is going to be CO2. If it's photosynthesis, the gas is oxygen. And so how much? Well, the faster you do it, the more you produce. The slower you do it, the less you produce gas. But that's how they'll try to get you. And obviously, they can ask you how much oxygen you produce. If I have more light or less light, how much oxygen am I producing? If I exercise more, I produce more CO2. What am I using more? Oxygen. So all this is, so now you lay your head down, Jonathan. Jonathan,
Yes.
Now you lay your head down. So you close the computer. I'm literally explaining it step by step. I'm literally explaining it step. I'm literally saying, this is what happens. And everyone should have already read the chapter, no? And I'm here after school on Wednesday and Thursday if you need tutoring. And there's tutors downstairs. I don't know what else to say. It's okay. You don't have to be successful, but I don't want you giving up. That doesn't make sense. Giving up does not make sense. All right. So when we deal with, that's the simple outline of what we're going to be talking about starting tomorrow, which is cellular respiration. We'll finish cellular respiration tomorrow, which is Wednesday. Thursday, we will finish photosynthesis. Friday, we'll go over the three different kinds of photosynthesis, and we're done with everything that's going to be graded on your final. That's it. All right. Now, if you're confused at all, I'm going to tell you what's going to happen to any future that happens to you, period. I don't care if you go to college or not. You go to the Army, Air Force, Marines, same deal. You go to work for your uncle, it's going to be the same deal. The only way you're going to understand anything is if you put effort into it. You have to prepare. You cannot walk into your first day at work at a factory where your uncle's setting you up with a great job and think that you're going to know what's going on. That's not happening. You have to figure out, you have to ask questions, you have to figure things out, you have to take notes, you have to re-ask questions, you have to make mistakes, you have to try again, you have to make mistakes, you have to figure it out. Just like in chess, just like in anything. Put the same effort that you put into things that you love, that you have passion for, into things you hate. Because it doesn't matter if you hate them or you love them. The only thing that matters is that what is it that you all chess players love to say? That we win. Right? Win. How do you win? You're successful. You learn it. You can forget it as soon as you're done with the class. Good for you. It's a waste of your time, I think, but that's fine. But learn it for the class. You got 4 more years after high school, college. The same thing's going to happen. And you think you're going to come into a college class and you're going to think, well, you think you're going to know the stuff the professor's talking about? You better know you're not going to know it. You better know you're not going to know it. And if you think they're going to explain everything in class, you're sadly mistaken. They cannot. It is impossible. Whether it's a religion class, or it's a business class, or it's a law class, or psychology class, there is no way in an hour they can explain everything. So you better start switching gears. You are in the big boys and girls club now. You're not little kids. Put the effort in outside the room. Prepare. I understand you're in high school. I understand you want to have fun. I love to see you have fun. All of you. You know I love your jokes. But it has to stop. You have to kick it into gear. When it's time to work, it's time to work. So, get it done, man. That's all. You don't understand this? Spend tonight reading the chapter. Watch a video on it at home. Come in tomorrow ready to discuss cellular respiration. Read the chapter. The work that's due that's outstanding is ridiculously simple. That's it. Ridiculous. It's a shame that I give you AP class. I record this and I'm okay with recording it. It's a shame that I'm giving you AP biology credit for those assignments. I should be ashamed of myself. I should go to my faculty meeting and ask for it. Stand in front of everyone and ask for their forgiveness. That I'm giving you such easy work. Get it done. But maybe giving you easy work gives you the wrong impression. I don't know. Get the easy work done. Read your chapter. Read it. You don't understand something, write the question down. Come in tomorrow, ask the question. Don't just sit there half asleep, think it, unless you know it already. If you know it already, that's fine. But you've got to break this stuff down. I could pull up Mayar's notes, and you can see the breakdown she does. You're like, I don't want to be a doctor. I don't care about biology. I hate biology. I don't care. You want the A. You want the GPA. You don't have to compete against Mayar or Alex or anyone else in the room. What you do have to do is you have a set of standards you have to know those standards so that you can be successful and do it even if you hate it. It doesn't matter if you hate it. I'm a religious person that took a philosophy class with an atheist professor who every day chose to argue against the existence of God and made fun of Christians. Openly made fun of Christians along with several other atheists in the room. What grade did I get in that class? A. Do you think I liked that class?
No.
I didn't leave that class with any friends, and I didn't care. I got my grade, I got my A, and I moved on. I learned the argument, even though I disagreed with the argument. And I argued against it with logic, because I had the strength and the knowledge to understand the argument that was being proposed to me. You all are intelligent enough to find those arguments, to understand these concepts. These concepts are not difficult. You can do it. You just have to find the energy and the motivation to do it. And if the motivation of liking it is necessary, change that mentally, do some meditation, see your religious counselors, see psychiatrists, I don't care who you see, change that, because you cannot depend on liking something to get it done. That cannot be. You have to get things done because they need to get done, because they are stepping stones to your goals. Your goals are what you need to get done. That's your passion. Your passion is where you want to go, what you want to get out of life. No? Is that correct? It's what you want. I don't care what that want is. That's your personal goals. But if you want those goals, you got to step through a lot of garbage. You have to jump over a lot of hoops. Start hopping. We all got to do it. All right, you got tonight. You know what you got to do. It's 2:53. Let's wrap it up, clean up. I'll post this for the people that are absent. Get the makeup work turned in before Thursday.